So implanted biomaterials and the foreign body response is depicted in this big schematic over time. So when the biomaterials are implanted into the body, like pretty soon, within seconds or within an hour, quickly the biomaterial will be coated by proteins, and that we call protein adsorption. And within 2 days, there will be an acute immune response. So that's the cells, immune cells will interrogate and try to attack this foreign body. The usual innate immunity is coming from neutrophils and then macrophages. And over weeks of the time, now these macrophages are trying to engulf, but they are frustrated. They merge and fuse to form this giant cell formation. And so it can take a week order. And also they, at the same time, they release a chemical substance such as interferon, interleukin, growth factors, which are secreted by these immune cells and having an effector on the other cells. So to signaling, so that's called a cytokine release. And over weeks of time, over like 3 weeks or longer, they tried to engulf, but they frustrated. So eventually what they do is a nearby fibroblast will secrete collagen to encapsulate this with a collagen capsule, which means if the biomaterials are inside the body, but effectively the body considers it kind of outside of the body by putting this collagen fibrous capsule. So this process can be depicted as a big, a nice figure here. I brought it. So implanted at this bare biomaterial surface will be soon absorbed by protein. And of course, acute inflammatory cells such as neutrophils are getting in. And the matrix deposition with a circulating monocyte gets in, turn them into macrophage, try to engulf it. And then nearby fibroblasts also try to secrete collagen fibers to fibrous encapsulation over like 3 weeks or higher time. And while these foreign body giant cells is formed to try to engulf this biomaterial. So that's the process can be explained as a protein adsorption within seconds to within an hour, recruitment of immune cells such as neutrophils and macrophages from the blood, and these macrophage fusion to form a foreign body giant cells on the order of kind of 2 to 10 days, and involving tissue-specific fibroblasts. Fibroblasts is those who secrete and generate fiber, collagen fibers, and also endothelial cells nearby blood vessel. And subsequent encapsulation of the implant by fibrous capsule. And these are usually avascular. And so here shows a little, another like electron micrograph morphology of biomaterial tissue interaction. So SEM is a scanning electron micrograph. So this is more like 3-dimensional, pretty high resolution electron micrography showing early thrombotic deposits of now heart valve material, which as an experiment exposed calf blood for just a few minutes. And it shows that this a star mark over here on the surface, you see this undulation. So this shows a protein adsorption. And the B is an experimental tissue reaction to relatively inert biocompatible material. And you show that this material M at the interface of this muscle tissue, you see this thin, very thin, only a few micron thickness, thin fibrous capsule has been formed. And C is similar to the previous one in H and E. I showed you that this dacron, which is biomaterial mesh, and nearby the granulomatous inflammatory response to the surgically implanted Dacron surgical suture. So that we call giant cells, and this granuloma as meaning that mass granulation tissue typically produced in response to infection, inflammation, or presence of a foreign substance. Granule means like small grains or rice grains or particles. And if we go back to this scanning electron microscope again, we see these RBCs, and there are platelets, which is a unactivated, and activated seems to have a lot more like branches coming out, and C is a clump. So I brought this picture of an unactivated platelet looks, and once it's activated, you see how striking their shapes are different. So next time we will try to understand the functions of biomaterial, which is involved in open heart surgery. And learning about the medical device, drug-eluting stents, now which is using to treat heart diseases. And thank you.